So today we are talking about the development of the cardiovascular system. This process starts at the 3rd week of gestation when the embryo cannot satisfy itself with enough amount of oxygen and nutrients. At this period of time taking place the gastrulation, and we know that at the 14th day of development we have the primitive streak, and in the anterior region of the primitive streak, the region which is nearby to the primitive node, there are located the progenitor cells for the heart field. From here the cells begin their migration, and they migrate to the lateral mesoderm, to the splanchnic layer of the lateral mesoderm. Exactly, they will reside in anterior region of the embryo, which is anterior most to the location of the neural folds. Here that will be the primary heart field. And from this region, from these cells will develop further the primary heart field, the chambers of the heart. Here we are looking at the lateral plate mesoderm, which have visceral layer, parietal layers, and in the lateral plate mesoderm, in the visceral layer, there is located this heart field, primary heart field, and these cells will be induced to via the underlying endodermal cells. They will be induced to form the myocardium, myoepicardial plate. So here develops the myoepicardial plates in the lateral mesoderm. Then under of it we have mesenchymal cells that will coalesce and they form endocardial tubes. These endocardial tubes will be surrounded by myoepicardial plate that arises from the visceral layer of the lateral mesoderm. The primary heart field initially is located in front of the oropharyngeal membrane, as we see here, that is the oropharyngeal membrane. So it is located in anterior most region to this oropharyngeal membrane. Then we also here we have to mention that we have also secondary heart field that originates from the neural crest cells that are located near to the hindbrain region. There is neural crest cells. They will migrate in upward direction towards the 3rd, 4th, and 6th the pharyngeal arches, and from there they migrate towards the heart tube, and they will add cells into the outflow regions, outflow tracts of the heart, and they will participate in the formation of these outflow regions, and they will also contribute to elongation of the heart tube in next stage. Then we have to mention here that formation of the heart tube, it takes place at the same time as the bilaterality is established. So the cells which form the heart chambers, they will have some order in right to left position, so that we will have laterality in the location of the heart. So for example, the cells of the right side of the secondary heart field will migrate to the left outflow tract, and from the left side they migrate to the right outflow tracts. So the laterality will be correctly established. And here we are looking that when the embryo folds, during the folding of the embryo that's taking place in cephalocaudal orientation and in lateral orientation. During the cephalocaudal folding of the embryo, the oropharyngeal membrane, it moves downward, and the neural tube is growing neural tube, that is brain vesicles will be, they will also move downward in ventral direction. By that way, the heart field will move first of all to the cervical region. From the anteriormost location, it will move to the first of all to the cervical region, and then it will replace to the thorax. So it will be its final location in the thoracic cavity, and oropharyngeal membrane will be located in front of it. Brain vesicles will located downward and in front of the primary heart field. That will be its final location. The heart tube, initially it has horse-shaped tube. It's the shape of horseshoe shape. And before lateral folding of the embryo, we have two tubes, in right side and in left side. Then during the lateral, as a result of lateral folding of the embryo, these two endocardial tubes will move toward each other, and finally they will fuse with each other, and we will have one tube, one heart tube that include endocardial tube, and these tubes fuse mainly in the central region, in caudal region, and the most caudal region of the tube will not fuse. Here we also can see that heart tube, it is inside, it becomes incorporated into the pericardial cavity. This one is pericardial cavity, and this heart tube becomes incorporated into the pericardial cavity. Initially, it is suspended in pericardial cavity via the dorsal mesocardium. The ventral mesocardium will not be formed, just we will have dorsal mesocardium. And its upper portion, middle portion will disappear and leave the sinus that will, pericardial sinus, that will, transpericardial sinus that will join, that will way of the connection between the right and left sides of the pericardial cavity. But in the caudalmost edge of the mesocardium will develop epicardial organ, and from this proepicardial organ will migrate cells that will surround this heart tube externally, and it will create epicardium. That will be epicardium, the third outermost coat of the heart. So we will have endocardium, myocardium, and epicardium. Myocardial cells produce extracellular matrix, that GLI, that is deposited between this myocardium itself and the endocardium. So we will have all of the three layers in the wall of the heart tube. And before talking about it, I want to show you the same processes by other illustrations to fix your knowledge. Here we are looking at the primary heart field, which is located in front of the neural folds. This is this one. And that one is showing us, this illustration is showing us that there are two heart tubes will be formed from the mesenchymal cells is formed endocardial tubes, and that is the myoepicardial cushions from which will develop the two heart tubes initially. Then that is the location of the primary heart field in front of the oropharyngeal membrane. And here we can see how the folding, cephalocaudal folding of the embryo lead to transposition of the heart field into the, first of all, cervical region, and into the, finally into the thorax. and that is a heart tube, endocardial tube, that begin to merge into the pericardial cavity, which is here, and finally it will be surrounded, that is a pericardial cavity, and these are two tubes just about withhold, two endocardial tubes. That is the stage before lateral folding, and because after the lateral folding we will have just one tube, endocardial tube, and here that is the inflow region with the line veins will carry blood into the heart tube, and that is will be inflow weight, and that is the outflow region where the blood will flow out into the aortic arches and into the dorsal aorta. So that we will have two dorsal aortas. They are paired, right and left, and the blood from this heart will flow into those vessels. Endocardial tube that is surrounded by myocardium, and that is a mesocardium, dorsal mesocardium. Here develops epicardial organ from which cells migrate and surround this tube externally and establish epicardium. That is develops in front of the larynx, as we see here. That is the larynx, and in front of it is developing heart. Now we are coming to the further development of the heart tube. That is the tube which just we mentioned, that is inflow way. This region of the heart, initial heart tube, is called sinus venosus. It has two horns, as we can see here. Then that region is initial atrium, primitive atrium. Then we have ventricle, that is dilated middle portion of the heart, and bulbus cordis. So that's the outflow way. And then we will have aortic roots here. We have to remember that aortic roots and most part of the bulbus cordis developed from the secondary heart field. It means that from the neural crest cells. And then as the elongation, so addition of the secondary heart field cells to the end of the heart tube lead to elongation of this tube, and elongation leads to the looping, to the bending of the heart. And finally we will see that this cranial portion of the heart tube bends caudally, ventrally, and to the right side, but this end, caudal end, it will bend in craniocaudal and in direction, and to the left side. Finally we will see the heart tube, which forms like loop-like region structure, in which this bulbus cordis bend downward, and it will downward, and then further the initial part of the bulbus cordis will participate in the formation of the right ventricle. This blue colored ventricle will be the left ventricle, and that will be atrium, initial atrium, and the sinus venosus will be located in cranial part of the heart tube. Now we are looking at this folding, at this bending of the heart. It's at the 28th day of development, and here, so that is the atrium, primitive atrium. This is ventricle, and bulbus cordis, initial part. So bulbus cordis we can subdivide into three regions. Initial region of the bulbus cordis, this one, this initial region here, we are looking here, it will form trabeculated part of the right ventricle. Then the middle portion, conus cordis. Conus cordis, it will form the outflow way from the ventricle, from the right ventricle, and truncus arteriosus. So truncus arteriosus further subdivides into the aorta and the pulmonary trunk. So that will be outflow ways of the heart tube. Then the atrium, here was the atrium. This atrium then will expand, it will expand, and as a result of expansion of the atrium, primitive atrium, this outflow tract of the heart from the right side will change its position into the middle location, the middle of the heart field. Why? Because of the expansion of the atrium. As a result of expansion of the atrium, so outflow tract, it means the truncus arteriosus, together with conus cordis, will move toward the center of the heart. Then here we are looking that, so that is a ventricle, it is a left ventricle. Here forms the right ventricle from the initial part of the conus bulbus cordis. And in their wall we can see trabeculation, so there will be trabeculated part of these ventricles. And between the ventricle, initially this is just one ventricle, and this bulbus cordis, here we have the ridge, the bulboventricular ridge, and it means the that is the inside of it we have interventricular foramen. This is between the right and left ventricles, and that is an arterioventricular canal that is that connects the lumen of the atria with the lumen of the ventricles. And here the right atrium will develop further, that will be the primitive left atrium. And then we have to mention about the something about the vessels, but before talking about it, let's look at bending of the heart tube in this illustration. That is sinus venosus, atrium, ventricle, and bulbus cordis, truncus arteriosus, and pay attention how, in which direction it bends. So it the outflow tract moves downward to the right side, and this atrium moves in cranial direction and to the left side. Finally, we will have this, that is a normal position of the heart. But in the dextrocardia, the bending of the heart tube will be in opposite direction. That is a problem, of course. And now we are looking how the heart chambers will communicate with each other. That is the sinus venosus. Here we can see that outflow tract of the heart tube. That is the atrium, then ventricle, and how the blood will flow inside of the heart tube. We can see the direction, and formation of the septa will subdivide the atrium from the ventricles, and finally then will form other septa about which now we are going to talk. Before talking about the septa formation in the heart, we have to mention that initially the sinus venosus, it has two horns. That is the opening of the sinus venosus into the heart, heart tube. How the sinus opens into the heart tube. Initially, we have to know that there is wide opening sinoatrial junction is enough. It has a large diameter. Then and here we can see two horns of the sinus, that is the right and that is the left horn. Into each horn open three veins: vitelline vein, umbilical vein, and common cardinal vein. It has two branches: anterior cardinal vein and posterior. And in this side is the same: vitelline vein, common cardinal vein, and the umbilical vein. And what can we see here? Here we have to mention them. Then at the 4th week there is taking place the shift of the blood from the left side to the right side, and as a result the left vitelline vein and the left umbilical vein, they undergo obliteration, and the common cardinal vein also undergo obliteration, and as a result the right vitelline vein becomes wider, and so the blood shifts to the right side, and that's why the right sinus horn becomes the only communication between the sinus venosus and the right atrium. As we see here, that is a right horn that will form the communication into the heart, into the right atrium. And from the left sinus horn only that remains, it will be just we will look here, it will be the cardiac coronary sinus and oblique vein. That will be oblique vein of left atrium, that is coronary sinus. That is only that remains from the left horn, and it will open into this vein which will form inferior vena cava, and the initial part of the sinus will be incorporated into the atrium. It will be incorporated into the atrium. Let's look here. Here we can see that the sinus, in the initial part of the sinus, it is incorporated into the atrium, into the left atrium, and finally that is the incorporation of the sinus horn, initial part of the sinus into the right atrium. So it will form a smooth walled region, part of the right atrium. This part, the primary part of the right atrium, becomes trabeculated portion of it, and the part of the sinus that is incorporated into this right atrium, it will form smooth walled part. Between them we will have crista terminalis that will be border between this smooth walled part and the trabeculated part of the right atrium. At the region where the sinus opens into the right atrium, there are two valves. It has two valves. That is the left valve, and this is the right sinus valve. Then the upper portion of the right valve disappears, but before it, these two valves, when the region where they fuse with each other, that is called septum spurium. That is septum spurium, where the right and left valves fuse with each other. Then, yes, that part, that right side of this valve will disappear, but its low region, this caudal region, this region will form two valves. It will be valve for inferior vena cava and valve for the coronary sinus. So again I repeat, they develop from the caudal, from the low part of this right valve of the sinus. They will form this valve for vena cava and for the coronary sinus. Then let's look at this valve. This is left valve, and left venous valve will, together with the septum spurium, they will fuse with atrial septa, with a secondary septum secundum. So they will fuse with septum secundum, and they will participate in the formation of the interatrial septa. So that is here. Now we are looking how the heart chambers will separate from each other. There are different ways of this separation of one chamber into some small chambers. First step here, we can see the formation of the thickenings in the walls, and actually in the wall of the heart we will see formation of these endocardial cushions. These are endocardial cushions that formed in the wall of the heart at the region, at the atrioventricular border regions, where the border between the atria and ventricles, there will develop these endocardial cushions. How they develop? Pay attention, that is endothelial cells that cover endocardium. These cells will undergo division and migrate downward, and they will produce extracellular matrix. As a result of deposition of extracellular matrix, this tissue becomes thicker, and this one will called endocardial cushions. So here, first of all, they will present at the region between the atria and ventricles. At these regions, we will have endocardial cushions. Pay attention, in this illustration we can see them. So we will have left endocardial cushion, right endocardial cushions, thickenings, and additionally we will have anterior and posterior, or cranial and dorsal cushions we will have also. And finally they will fuse with each other, not fuse, they will move toward each other, and finally between them will be formed channel, and by that way these cushions will subdivide the heart into atria and ventricles. Also, the cushions will participate in the formation of the interatrial and interventricular septa. Their membranous portions also will develop from these endocardial cushions. So in the atrioventricular endocardial cushions will participate in the formation of the anteroventricular canals. Also, their free edges will form the valves, atrioventricular valves. Also, other cushions we will have at the bulbus cordis, at the conus, at the truncus arteriosus. There also develop endocardial cushions that will subdivide one outflow tract from the heart into two parts, the aorta and the pulmonary trunk. Another way how the heart chambers subdivide into two, it may be by other way. For example, we have here one chamber. If one chamber subdivides into two and both of them expand, and the stripe, the narrow stripe between these two parts, it stops its division and its growth, and what it will be resulted in. Both these parts, they expand, but this middle portion do not grow. As a result of this expansion, their walls will fuse with each other, and finally there will be formed septum. It is how the septa in the interatrial septa will develop. They will develop by this way. So they will subdivide to atria, right and left, from each other. Now we will look how these septa are developing. Here we can see the left atrium and the right atrium. Initially, in the 4th week of gestation, from the roof of the atrium starts to grow crescent shape, crest-like structure. That is the septum primum. That is the accumulation of cells that move downward. It is septum primum. Then, as a result of growth of endocardial cushions, the cells of endocardial cushions move upward, and finally they will fuse with the free end of the septum primum. So septum primum temporarily will separate two atria from each other. Then in the proximal part of this septum primum, we will see death of cells. Finally, they will form opening that is called, if pay attention to the septum primum, under of it will form ostium primum that then will fuse. Then in the septum primum, in proximal part of it, there is formed there is ostium secundum. So that will called ostium secundum, this opening. Then as we know, the sinus venosus, its proximal part, becomes incorporated into the right atrium. As a result of this incorporation, there was ostium spurium. This spurium will fuse this ostium spurium, and together with the they will form together with the downgrowth, another downgrowth that is formed from the roof of the atrium. There will form septum secundum, that is blue colored. So we will have septum secundum that grows downward. Its free edge, pay attention, its free edge, and here it has free edge. Between these free edges of the septum secundum will be formed another opening that is foramen ovale. It will have oval shape. That's why this opening will called foramen ovale. And we know that there is ostium secundum is located. So the remaining part of the septum primum will be opposed to this foramen ovale, and it will serve as the valve for this foramen ovale, and the blood will shift from the right side, to the left atrium via this foramen ovale, and the septum primum remaining part of the septum primum will be the valve, which will close, and it will form the like flip that is the valve for this foramen ovale. And then at the same time we can see cushions, how they grow and how they form the valves. Here the valves are formed, and they are also formed from the endocardial cushions. The cushions also will form the membranous part of interventricular septa. Here we can see it. And in this one we are looking how the endocardial tube, it is inside of the it becomes bulged into the pericardial cavity, and that is here we can see the foregut, and that is so neural tube here, then the foregut, and the endocardial tube, initial endocardial tube. That is inside of this pericardial cavity, and the dorsal mesocardium here, as we know that in the caudalmost part of it there is formed proepicardial organ, from which cells move, migrate, and surround this heart tube to form epicardium. Pay attention to this illustration. Here we are looking how there is septum primum is formed. Inside of the septum primum, some cells of dorsal mesocardium, actually dorsal mesocardium, it has neural origin, it has developed from the neural crest cells. So these cells together with septum primum, then they invade into the heart, they enter like into the heart, and they will grow into the veins, pulmonary veins. So they will form pulmonary veins. The remaining part of these cells will move at the tip of the septum primum towards the endocardial cushions. And so their pulmonary veins are formed, and their initial part also will be incorporated into the left atrium, like sinus venosus incorporated into the right atrium. Here we will see incorporation of the initial part of the pulmonary veins into the left atrium. They will form here the smooth-walled region of the left atrium, and the remaining part will be the trabeculated portion of it. And so that is smooth-walled part of the left atrium, that is smooth-walled part of the right atrium, and so all of the structures we just mentioned, that is how pulmonary vein is developing here, and its initial part is incorporates into the left atrium, and by that way it expands the lumen of that atrium. Here we can see how the endocardial cushions develop, and actually in the atrioventricular ostium we have 4 endocardial cushions, right and left, this one lateral cushions, right and left, and here we are looking at the superior and inferior cushions. They move toward each other, and finally they subdivide this just one opening, atrioventricular opening, into 2 parts, right and left atrioventricular canals. Initially, when the blood will flow through these canals from the atria to the ventricles, they will lead to thinning of this wall, and finally, pay attention, this mesenchymal tissue here at the atrioventricular canals, when all the blood will move through it, this wall becomes thinner, and finally the valves are formed, and they're trabeculated part of the myocardium, myocardial cells, and some of the cells here will be converted into the connective tissue, and here will be formed chordae tendineae that connect papillary muscles at the bottom of the ventricles with these valves. Then here we are looking how the so that is formation of the right and left atrioventricular orifices. Here we also can see the other cushions that are formed in the truncus arteriosus, that is superior, the right superior truncus swelling, left ventral conus swelling here, then we will have right dorsal conus swelling and left inferior truncus swelling. All of these swellings will move toward each other, and finally they will subdivide this one lumen, one tube, into 2 tubes. One of them will be pulmonary trunk, the other one will be the initial part of the aorta, and this septa will have spiral way. And so we have to mention here that the pulmonary trunk, that is this one, and that is aorta, between them will be formed the ductus arteriosus that connect these pulmonary arteries with the aorta. So the blood from this pulmonary trunk, or some of the blood, will flow into the aorta. some of it will move to the lungs, but because lungs are infunctional, most of the blood moves to the aorta, to the aortic arch. Then let's come to the, yes, that's how the this truncus and ductus arteriosus subdivides. Pay attention to the right conotruncal ridge and left conotruncal ridge. It's inferior, it's located uppermost region, it's more inferiorly. This ridge creates cells that move downward and to the right, and this ridge creates cells that move downward and to the left. So they will subdivide all of this tube into two. So conotruncal septum is formed. That is, the pulmonary channel will be here, that is outflow from the right ventricle, and from the left ventricle it will be aortic channel, as we see here. So this truncus subdivides into two. Then we are coming to, yes, pulmonary valves also develop here. So the conotruncal swellings, they they subdivide this lumen into two channels. At the same time, they create the valves in the entrance of these large vessels. So here we will have these valves. So we have to know that the development of the truncus arteriosus, its main source of development, that is the secondary heart field. And secondary heart field, it is the source of it, that the neural crest cells. That's why if there is a problem, genetical problems in the secondary heart field cells, it may lead to different defects in the formation of the bulbus cordis and the truncus cordis, then the arteriosus. So outflow tracts of the heart will suffer, and there may be different pathologies. For example, tetralogy of Fallot. Tetralogy of Fallot, it has complex of problems, defects. First of all, that is a pulmonary stenosis. Then there will be the opposite position of the large vessels. Then there is also we will have here we can see patent ductus arteriosus that remains after the birth time, and the stenosis of the aorta may be initial part of the aorta. So that is the tetralogy of Fallot, that is a result of defects in the secondary heart field development. Besides it, we have also persistent truncus arteriosus. We may have persistent truncus arteriosus and transposition of the great vessels, because neural crest cells also contribute to much of the face craniofacial region of the embryo. So this tetralogy of Fallot will also associated with different defects in the face of the embryo, that we also have to not to forget. Here that is a secondary heart field cells, migrating cells that come towards the heart tube and to its outflow tracts. And also we have to mention the development of the interventricular septa, how they develop. Actually, the muscular part of interventricular septa, they develop as a result of expansion of the ventricles, and the mid portion of these ventricles, they will fail to expand, to develop. That's why two large cavities then will merge and will fuse in the midline, so that will be formed the muscular component of the interventricular septa. The membranous part, the membranous part of the here we are looking at the initial septa, initial ostium, which connect two ventricles. There is interventricular ostium and muscular interventricular septum we can see here. Then from the endocardial cushions, the cells that migrate, they move downward, then they will fuse with the muscular wall, and that will form complete separation of the ventricles, the right and left ventricles from each other. Here we can see the septum secundum, the foramen ovale, and that is a valve of the foramen ovale which is formed by the septum primum. And also that is the valve for the vena cava. That is valve of the vena cava. Then we have to know about the development of the conducting system of the heart. As we know, the heart undergo contraction under the influence of impulses that arise within heart itself. There are atypical cardiac muscle cells which form this system, and we know that there is the atrioventricular. We have sinoatrial node, which is located actually initially sinoatrial node. It is located at the bulbus cordis, at the conus truncus, at the truncus region. Then as a result of looping, as a result of formation of the heart chambers, and especially as a result of incorporation of the bulbus into the right atrium, and then that region, that part of those cells which were located in the wall of this bulbus, they will be now located at the region of the right atrium, near to the opening of the superior vena cava into the right atrium. There will be located sinoatrial node. Then we have another node which is then further will be located in the interatrial septa, that is atrioventricular node. The atrioventricular node cells will take their origin from two sources. First of all, from the caudalmost part of the sinus venosus, and also some of the cells of the interatrial septa. So cells together, myocardial cells, together will form atrioventricular node and atrioventricular bundle. So this will be the conducting system of the heart, which will then further, it will generate impulses for contraction of the heart. Now we are coming to the formation of the valves in the wall of the large vessels. Here we also have almost the same mechanism as in the atrioventricular valves. Here we also have cushions. These are conotruncal cushions, right and left. Then we have these minor truncus swellings, and they together will form the walls of these large vessels. And in the walls we will see thinning regions. They will form semilunar valves. Semilunar valves that will located in the entrance of these large vessels, and they will prevent backflow of the blood. Now we are coming to the arterial system, to the vascular system. In the vascular system, so here the heart, what we have in the embryo, and so we can see the development of the vascular system in the embryo. And here we have the yolk sac, that is the embryo body, and that is a heart tube which have sinus venosus and the heart itself. To the sinus venosus come veins. Which veins we have in the embryo? First of all, these are vitelline veins which come from the yolk sac. Then we have common cardinal vein, which carry blood from the body of the embryo, that it has posterior branch and anterior branches. Then we have umbilical veins. So umbilical veins also carry blood into the sinus venosus. Then we have arterial system. And the arterial system, blood vessels of the embryo develop by two ways: vasculogenesis from the blood islands and angiogenesis. Angiogenesis is sprouting from the existing blood vessels. But vasculogenesis develop two dorsal aortae and also large cardinal veins. All of other vessels develop from the via angiogenesis. Also the aortic arches also they take the origin via vasculogenesis. Initially from the heart arises the aortic sac. Aortic sac have some subdivides into the aortic arches, and they reside within the pharyngeal arches. So each pharyngeal arch, it has, as we know, 6 pharyngeal arches develop in the embryo, and inside of each of them there is the aortic arch also. This aortic arch then join the dorsal aorta, left and right side. Then this aorta, here we can see dorsal aorta, and from it we will see branch, vitelline artery, and then the umbilical arteries. Then these vitelline arteries will change their position, and they will further will develop into celiac artery and superior mesenteric arteries. And from the umbilical artery will develop inferior mesenteric arteries. Now we are looking at the aortic sac, and these are arches, aortic arches, which carry blood into the dorsal aorta, right and left side. Here we can see. And then we will see some further development. There will take place some change in the wall. That is the aortic sac, from which arise these aortic arches, and they join dorsal aorta. Further in development, and we have to know that we cannot see all of these aortic arches at the same time. When the proximal ones are formed, we do not have the distal ones. When the distal ones formed, some of them disappear, some of these aortic arches are disappeared. So the first aortic arch and the second aortic arch then will disappear, and first aortic arch will remain as the maxillary artery, here we can see the maxillary artery, which is a remnant of the first aortic arch, and then further in development the second aortic arch also will disappear, and they will persist as hyaloid and stapedius arteries. We have to mention that these aortic arches that develop inside of the pharyngeal arches, there and we know that material of mesenchyme for pharyngeal arches, it develops from the neural crest cells. That's why the endothelial cells, then the all of the components of the vascular wall, smooth muscle cells, and connective tissue elements, they take origin of this aortic arch. They take origin from the neural crest cells. And then we are looking at speaking about the aortic arches. We have to mention that initially we have just one entrance, that is aortic sac. But when the aorticopulmonary septum subdivides the truncus arteriosus into two parts, pulmonary trunk and the aorta, then we will see some change in this arterial system. Exactly speaking, the aortic sac will subdivide into two horns. It will be the left and the right horns. From them, the right horn, and they will have some not exactly symmetrical way of development. In the left side, it will be aortic arch, and in left side we will have brachiocephalic, and then it will continue into the right subclavian arteries. So that will be the horns of the aortic sac. Then we will see some change in the vascular system because of this shift of the blood. Here, that first arch, as we see, it disappeared. The second one also. Third one will persist, and this third one will form common carotid arteries, and so the third one will develop into common carotid artery and into the internal carotid artery. The remainder part of internal carotid artery will develop from the dorsal aorta, from the right dorsal aorta, remaining part of the dorsal aorta. And the external carotid arteries, they will be sprouting from the third arch, that is external. Then the fourth one, as I mentioned, the fourth aortic arch will develop into the right subclavian in the right side and into the aortic arch of the aorta in the left side. And then this subclavian arteries remaining part develops from the remaining part of the dorsal aorta, as we see here, the right dorsal aorta, and its remaining part will continue as continuation of the right subclavian artery, and also some part of it will develop from the seventh intersegmental artery. That is the fourth. Then the fifth aortic arch, it never develops, or it is rudimentary, it disappears just after the development. And finally the sixth one will form the pulmonary arteries, the right side and in the left side. And in left side, actually in the right side, this aortic arch will lose its contact with the dorsal aorta, and it will pulmonary artery which go to the lungs. And in the left side, we will see the continuation, the connection of the sixth arch, that is the left pulmonary artery, with the aorta, dorsal aorta, remaining part of the dorsal aorta, that is as ductus arteriosus. It will persist. And then we will see, as a result of cephalocaudal and lateral foldings of the embryo, we know that heart changes its position to the thorax. It comes to the thorax, and finally the extremities, upper extremities, will located above the level, something above the level of the heart. That's why the brachiocephalic arteries, subclavian arteries will move upward, and they change their place of origin from the aorta, from the level of seventh intersegmental artery to the level near to the common carotid artery, and to the common carotid artery, as we will see here. Subclavian artery and common carotid artery, they will have the same level of origin from the aorta, and in the left side the same. So the position of the subclavian arteries will come upward, and also common carotid arteries and subclavian, they undergo elongation as a result of change of the position of the heart to the thoracic cavity. And as a result of these processes, as a result of elongation of these vessels and the transposition of the heart, we will see the changing position of the nerves. That is the laryngeal nerves, which are branches of the nervus vagus. If initially these recurrent laryngeal nerves, they are located at the level near to them, this pulmonary arteries level, as a result of the ascendance of these vessels, they will also change their position to the upward region, and we know that these nerves will then innervate larynx, and these vessels or these nerves will hook around this right brachiocephalic artery. But in this side, in the left side, because truncus arteriosus persists, so this connection in this side, it's lost connection. But in this side, because of not lost connection between the pulmonary artery and the aortic arch, this nerve here will hook around the arch of the aorta. That is also we have to mention and we have to know. So finally, we can see derivatives of the aortic arch. So first aortic arch persists as maxillary artery, second one as hyoid and stapedial arteries, third one will be common carotid and first part of the internal carotid arteries, and the fourth left side arch of the aorta from the left common carotid to the left subclavian arteries. Right side, it will be right subclavian artery, proximal portion. Sixth one, it will be left pulmonary artery and ductus arteriosus. It is in left side, but in right side, it will be right pulmonary artery, just we saw in the illustration. And so which change will take place besides mentioned, all of the mentioned changes? We have to know here that the dorsal aorta, part of the dorsal aorta that is located between the third aortic arch and the fourth aortic arch, that were called carotid sign, the carotid duct, this region will disappear. This one, in both sides, this carotid duct will disappear. Then the part of the right dorsal aorta that is located after the level of the seventh intersegmental artery, it also will disappear. And in the left side, the dorsal aorta, the distal part of the dorsal aorta, it will persist, and it will form here, it will form the aorta, that is a dorsal aorta, which will carry blood to the organs of the abdominal cavity and low extremities. And here we also can see how the aortic arch and derivatives develop. So actually, as we see here, the first and second aortic arch will disappear, and there will develop just third, fourth, and sixth one. Fifth one also will not develop actually. And from the third one develop common carotid artery, and its branch will form internal carotid artery. That's remaining part of it develops from this dorsal aorta, remaining part of the dorsal aorta. And then truncus arteriosus that connects the pulmonary artery, left pulmonary artery with the aorta, will also persist. And the dorsal aorta, as we see here, the region of it between the third and fourth pharyngeal aortic arch will disappear, as we see here, and its cranial portion persists at continuation of the carotid arteries, and the low portion persists at the right subclavian, and in left side it will be the aorta. So that is the dorsal aorta that will persist and will go downward. Here we also can see how the aortic arch develop and which vessels develop from them, and the recurrent laryngeal nerves how they will ascend their position and how they will hook around the blood vessels. In right side it will be the subclavian artery, and in left side it will be dorsal aorta, that's arch of aorta, because of the persistence of truncus arteriosus. And in some cases the ductus arteriosus will persist, and there are some substances which will lead to constriction, which assist in constriction of it, that are the indomethacin that closes the duct, but prostaglandins, they keep open the lumen of the ductus arteriosus. In some cases we have also 0.5% of people may have aberrant right subclavian artery. It is usually asymptomatic, that we also have to know. And there may be double aortic arch, that's also some of malformations, and through this will pass the trachea and the esophagus, and the arch of aorta may be subdivided into two parts, and from them will flow blood. That is also one of. And coarctation of the aorta, here we can see that's the contraction region, that's actually, and the thin region may have long length, so that will cause problems. And which arterial vessels actually have the embryo when it is 4 millimeter in length? There are, as I mentioned, there are large vessels, dorsal aorta, and from the dorsal aorta we have vessels. These are vitelline arteries. They take the origin from the dorsal aorta and carry blood to the yolk sac. Then these vitelline arteries will develop into the celiac and superior mesenteric. And we have also umbilical arteries that will develop into the inferior mesenteric arteries. And all of these arteries, celiac, superior mesenteric and inferior mesenteric arteries, they will supply the gut, foregut, midgut and the hindgut. About the umbilical arteries actually, they have initially they come from the aorta, they branch from the aorta and carry blood inside of the umbilical cord along the allantois. They go to the placenta. Afterward, they receive additional branch from the aorta, that will be common iliac arteries, and they will lose the initial contact from the dorsal aorta. So the blood from to these umbilical arteries now will come via the iliac arteries. Then when the obliteration of the umbilical arteries will take place after the birth time, the distal part of the umbilical arteries will develop into the ligament, into the umbilical ligament, but the proximal part will remain as vesical artery and internal iliac arteries. So these are further developments for these arteries. And how the carotid arteries develop? Carotid arteries, they develop in the wall of the heart, from the epicardium, from the epicardial ridge actually. They develop from the epicardial ridge, as we know. In the wall of the heart, initially we have this mesocardium, dorsal mesocardium, that is the bridge that connect the heart tube with the pericardial cavity. In this region develops a proepicardial organ, and proepicardial organ, its mesenchymal cells, they originate from the secondary heart field. It means that they are neural crest derived cells. Then these cells will they migrate and they form the coat around the heart tube, and they form epicardium around the myocardium. Then, in the wall of these epicardial cells, among epicardial cells, neural crest cells develop into the mesenchyme, and from the mesenchymal cells will develop the wall of the coronary vessels, endothelial lining, smooth muscle cells in the wall of these coronary vessels. So coronary vessels develop inside of the epicardium. Then their initial region, it will invade into the aorta, and finally we know that origin of the coronary vessels, it's from the initial part of the aorta where this vessel originates from the heart tube, and in the initial part of the heart tube of aorta will arise the coronary vessels that will supply the heart tube. So that's the end of this lecture. Thank you for attention. See you on next lectures.